All right, guys, so before we actually get started on Unit 2, my next slide is going to be a repeat of a slide from Unit 1 because I missed a pretty big point on there that you guys should know. So I'm going to go to that next. We'll cover all that, and then we'll get started on Unit 2. All right, so turn to page 4 if you have your book. We're looking at the autonomic component of the nervous system again. So just as a small review, we said at the very top, we said parasympathetic is going to release ACH at both the pre and postganglionic synapses, right? What makes it a cholinergic system is what is released at the postganglionic synapse because that is what is hitting the target cell, what the postganglionic neuron releases. So parasympathetic, postganglionic releases ACH. That makes it a cholinergic system because the ACH is reaching the target cell. If we look at sympathetic further down, preganglionic is going to release ACH, but the postganglionic releases norepinephrine generally. That makes it an adrenergic system because the postganglionic neuron releasing norepinephrine means that the target cell is receiving norepinephrine. It's an adrenergic system. Now, right under that, we have an exception. I put it in pink and bigger writing on my slide because you absolutely need to know this for the exam. This is the exception where postganglionic sympathetics release ACH at the target cell instead of norepinephrine. So make sure you know this. Sweat glands and erector pili muscles are receiving ACH at the postganglionic synapse. So in the red box to the right, I typed up some questions real quick as a review of this whole slide. Looking at the second one down, FIB stands for fill in the blank. So he could ask you a question as a fill in the blank as easy as which target cells are ACH released at for postganglionic sympathetics. Those are the two examples, sweat glands, erector pili muscles. You would write those two as your answer, and there's your points. So easy points if you know it. I wouldn't be surprised to see this on your exam. I should say I'd be surprised if you didn't see it on your exam. So definitely make sure you know it. Going to the top, I guess we can go through these questions together. Which of the following releases ACH at the target cell? So A, B, and D, those are our classic examples of it says postganglionic sympathetic. So we're thinking norepinephrine, and then the examples, glandular epithelium, smooth muscle, cardiac musculature. If you look at the top of the slide, that's the target cells that we listed. So those won't be the answer because they release norepinephrine there. Preganglionic sympathetic for letter C, that wouldn't be a good option because the question is asking which of the following releases ACH at the target cell. So the preganglionic sympathetic doesn't reach the target cell. It synapses on the postganglionic, which then reaches the target cell. So then we have E, postganglionic sympathetic at sweat glands. So that is our classic example of the exception where ACH is released at the target cell. So I hope that makes sense. Skipping down to the next multiple choice question, which of the following does not release ACH? So this isn't asking specific to a target cell. It's just saying which of the following does not release ACH. Somatic efferent does reach the target cell. That reaches our skeletal muscle, right? And that's going to release ACH. So that won't be correct because the question is asking, which of the following does not? Preganglionic parasympathetic, postganglionic parasympathetic. We know those both release ACH. There's no exceptions there, so those won't be right. D, preganglionic sympathetic. Looking on the slide to the left, we know that is going to release ACH. Postganglionic sympathetic releases norepinephrine generally. So that would be our correct answer. F under that is the exception, sympathetic at erector pili muscle. So that is where we do release ACH. So that's a good example of how a question might show up on your exam as a multiple choice. It requires a bit of thinking, but like I said, if you remember postganglionic sympathetic, that's the one that is the exception. or I should say that is the one to memorize that releases norepinephrine, and everything else is going to release ACH. Which of the following is an example of an adrenergic system? So it's another way he could ask the question rather than asking which of the following releases norepinephrine. He can use that other term to describe the same concept. So for this question, parasympathetic to glandular epithelium, we know that's ACH being released there, so that would be cholinergic. That's wrong. Same thing goes for B, somatic efferent, that releases ACH. That's cholinergic. C, sympathetic to sweat gland. That's the exception where ACH is released instead of norepinephrine. So that would be cholinergic as well. D and E, those are classic examples of postganglionic, sympathetic, releasing norepinephrine. So that would be the correct answer, D and E. Hope that makes sense. If you have any questions on that, just let me know. So, TLDR, too long, didn't read. Postganglionic sympathetic releases norepinephrine. Everything else releases ACH. So the examples for the postgang sympathetic releasing norepinephrine, that's our glandular epithelium, smooth, and cardiac muscle. If the example isn't that, postganglionic sympathetic gland epithelium, smooth, or cardiac muscle, it's going to release ACH. Okay, now on to Unit 2. I know I feel like I'm beating a dead horse here with that whole explanation, but my goal is that hopefully you guys are like, oh god, she won't shut up about it. We get it, and that means that you are learning it and that you're going to be getting these questions right on the exam. So, moving on to Unit 2. Unit 2 starts on page 9. We're looking at embryology and histology of the nervous system. I'm going to be honest, a lot of this section is straight memorization. I'm a super visual learner, so it's not my favorite. I like when we get into all the testable diagrams and whatnot, and you can really visualize what's going on in the content we're learning. A lot of this stuff is just memorization, so just to let you guys know, and sorry about that, I guess. So looking at this first page, that's pretty much what we're dealing with, is memorizing this chart, more so at the bottom. The first half of it, going through week 1, week 2, week 3, I wouldn't worry too much about all of this showing up on the exam. If I'm going to pick something from that first half to know, I would say know what I've got on this slide. Week 2 is the bilaminar disc formation. That's when you have 2 layers. It's nice that bi means 2, right? So week 2, you have the bilaminar disc and it's 2 layers. So they all got 2 in there. I would also know week 3 is the mesoderm formation, and that's the 3rd germ layer. So again, it's nice that week 3 we're having the 3rd germ layer form. And I think there's some overlappage in embryo for this content, so that's kind of nice too. You should be familiar with it by the time you get to the exam. Looking at the last half of the chart where we see all these arrows kind of splitting up into different sections here, this you're going to want to circle and you're going to want to memorize this. So we have 3 primary brain vesicles in development. And then the 3 primary brain vesicles, those are prosencephalon, mesencephalon, and rhombencephalon. Those develop into secondary brain vesicles. And then the secondary vesicles develop into our mature structures of the brain. So that's what this chart is showing. You got to memorize this chart. I'm sorry, it's tedious, but there's usually a few questions on the exam from this part right here. So if you look down at the bottom portion where it says week 5, 6, those are all the mature structures that develop from the secondary vesicles. The reason I say this is just pretty much straight memorization is because this is pretty much everything we're going to learn about this entire course. Each section of each unit here on out is going to be dedicated to a different secondary vesicle. If you flip through your book, you'll see there's a section on the telencephalon, there's a section on the diencephalon, on the mesencephalon. So I'm not going to go teaching you all that stuff because it's just too much. It's just an overview of the development and what the mature structures are that we're going to learn about. I do have up on my slide though a helpful way to know what primary brain vesicles develop into the secondary. So first we have TED-D is a pro. So breaking that down, that can help you remember the prosencephalon forms the telencephalon and diencephalon. So the word by itself, pro, that's the primary. Moving on to page 10, there's a whole bunch going on with these diagrams and figures. These are not testable diagrams, so they will not show up on your exam. So don't worry about memorizing the labeling and everything. Understand what's going on in these figures. It's showing the development and flexures, and the figure 2 is actually, if you look at that chart we were talking about on the last page, that kind of helps put it into a picture for you. So it's showing all the primary and secondary brain vesicles. So if that helps you memorize what's going on that last page, then look at that. But everything that you should absolutely know for the exam from this page, I have written up on this slide. If you don't have this stuff in your book yet, write it in the book. Make sure you know it. So we have 3 germ layers, right? Endoderm, mesoderm, ectoderm. Big thing to make sure you know is that the ectoderm is the germ layer that forms the nervous system. So multiple choice question, which germ layer forms the nervous system? You could have all 3 listed there as answers. Ectoderm is your correct answer. So absolutely know that. So I have up on the slide, ectoderm becomes the neural tube and the neural crest. So that's what's going on in this figure 3 here. Important thing to know here is that the neural tube forms the CNS structures, which is our brain and our cord, right? Talk about that. I think it was like on the second page of Unit 1. Neural crest is what forms the PNS structures. So tube, CNS, crest, PNS. An easy way for me to remember that the tube is what forms the CNS structures is if I picture a slim tube, it kind of resembles the spinal cord to me. So then I know the cord is part of the CNS. That's what helps me remember. Tube forms CNS structures. And then crest is everything else. So as soon as we leave the spinal cord or brain in the nervous system, we're talking about the PNS. One more time. Ectoderm forms the nervous system. Ectoderm forms the tube and the crest cells. Tube forms CNS. Crest forms peripheral nervous system. Okay, moving on to figure 2. So we have our cervical flexure and our cephalic flexures in bold on that very first figure there, and the very first image. Big thing you got to know about these is that the cephalic flexure is what forms our cervical curve. Our cervical flexure disappears later. So don't get confused on that. Some students might want to think, oh, cervical flexure forms the cervical curve. That's false. Hey, I just made a good true-false question, which I don't know if I mentioned. He does do some true-false on your exam. So he could say something as simple as true or false. The cervical flexure in development is what forms the cervical curve. That would be false because the cephalic flexure is what forms the cervical curve. I guess I should put that, I guess I could make a quick true-false example and put that on this slide. So I'll do that. But those are the main things to know on this page. If you don't have them written down or highlighted, make sure you do that. Now we're on page 11. So some numbers to make sure you're aware of at the top. Brain at birth is going to be 10% of our body weight and uses 60% of oxygen. So I think Dr. Torgerud mentions this in class. When you look at a baby, newborn, their head looks like way disproportionate to the rest of their body. It looks huge. So it should make sense that brain at birth is going to take up more percent of the body weight compared to if you look at the brain as an adult, it's going to be 2 to 2.5% of body weight. We kind of grow into our heads. Looking at percent oxygen use, it's also going to decrease. So we go from 60% at birth to 20% as an adult. Make sure you're aware of these numbers. Should make sense that they decrease as we develop. Another important point to know, birth to 1 year, the brain more than doubles in size. So it says in the book 2.5 to 3-fold increase. Highlight that, star that, make sure you're aware of that as well. All right, so on page 11, moving along, we're now on the histology of the CNS. So this is basically the cell types in the central nervous system. I recorded myself going through all of these through the book, and I realized I am pretty much just telling you guys the important terms to know for studying for the exam. So instead, I have a chart that I left in Davenport, so I'll post it in a couple days when I'm back and I can get it. Pretty much puts all of the information you need to know for the exam on one page. So if you study that, you should be okay. I ended up typing a lot of it out here, so I'm not just gonna talk through the book. I'm not really adding a whole lot of information, so just let you guys study that, and that should be good. I will say a few things about the cells of the CNS. So the chart on page 11 shows the neuroepithelial cells. Those are, you can think of those as the stem cells. So if you don't have that written down, I would write that down. And I have it on my slide here too, because they're going to differentiate into more specific cell types, which are the neuroblasts, ependymal cells, and glioblasts. So those are all the derivatives of the neuroepithelial cells. Looking at the glioblasts, you can see those further differentiate into astrocytes and oligodendrocytes. So, and it's listed under the glioblast cells as well. I would write it in the chart too. Glioblasts can undergo mitosis. So make sure you know that. I've got it listed up here on my slide as well. That's different than neuroblasts, which if you look at the chart, neuroblasts, there's an arrow that goes down to neurons, which might make you think that they are going through mitosis to become neurons, but actually it's going to be more of a development maturation into neurons. So neuroblasts aren't capable of mitosis like the glioblasts are. If you see the word mitosis, think glioblasts. If you see develop or mature, think neuroblasts into neurons. A couple simple tips just looking at this slide here. I'm not looking at the book at this point because I've transferred most of it to the slide, but astrocytes are going to be the most numerous cell type. Astrocyte starts with A, so does abundant, so that was an easy way for me to remember astrocytes are the most abundant cell type. They're important for the blood-brain barrier as well. Way for me to remember that was astrocyte starts with A, BBB, or blood-brain barrier, is the next letter in the alphabet. So A, B, 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 I would just tell myself when I was studying. And again, I know it's silly, but it does help you remember it, or at least it did for me. So if it helps, great, use it. Jumping back up to 2 on the slide, glioblast cells, we said they're capable of mitosis, and they give rise to astrocytes and oligodendrocytes. Make sure you know the 2 cells that they give rise to. That sounds like it could be a good fill-in-the-blank question. Which 2 cells do glioblasts give rise to? And then you would have to write down astrocytes and oligodendrocytes. Astrocytes and oligodendrocytes have different types that you need to know. So looking under astrocyte, you have protoplasmic, and that is found in gray matter, and fibrous, which is found in white matter. You have to know the types, and you have to know whether it's found in gray or white matter. So make sure you're aware of that. Oligodendrocytes, they're all new similar words. We've got perineuronal and protoplasmic. So oligodendrocytes are the perineuronal. Those are also found in gray matter, and those are involved with nutrition. Interfascicular oligodendrocytes are in white matter, and those are what form myelin in the CNS, and we're going to talk a lot about that. So you're going to know interfascicular oligodendrocytes like the back of your hand by the time the exam comes around. But one thing I noticed that helped me remember this for the exam, so we've got protoplasmic astrocytes are found in gray matter. Perineuronal oligodendrocytes are found in gray matter. Both of those start with P, and both of them are found in gray matter. So that both starting with P helped me remember that those were the ones that are found in gray matter. So if that helps you remember that, great. Taking it one step further for the oligodendrocytes, perineuronal, if you see on the slide here, I capitalized the N's because that is like a reminder for me that the perineuronal are involved in nutrition. So again, something silly that helps you remember it for the exam, all these new weird words that you might not have ever seen before. So you gotta kind of do what you gotta do sometimes to remember these things. So hopefully that helps a little. We're gonna hit ependymal cells and the ventricles pretty hard by the end of this exam material, so I'm not gonna go over that. But you guys are responsible for this information. Once I get that chart posted, that'll be super helpful because it'll have everything that has to do with the histology on one page. One more point on this section, the red box I've got here on the right says astrocytoma, glioblastoma, ependymoma. So if you have oma at the end of a word, it means tumor of. So these are tumors in the brain. And these are easy points on the exam if you know them. Something as simple as either a fill in the blank or a multiple choice question. Which of the following is the most common primary brain tumor? Which of the following is the most lethal brain tumor? So make sure you're aware of these. I will say, and I put it in red, astrocytoma is the most common primary brain tumor. Astrocytes are the most numerous cell. So that should make sense then. Ependymoma is going to be the most restrictive. That is in pink there. And I have under 5 listed that the ependymal cells line the central canal and ventricles. I think by the end of this exam information, this is going to make a lot more sense to you guys, but the central canal is a very tiny channel in the cord, and the ventricles are in the midline of the brain, also small areas. So to me, it makes sense why ependymomas are most restrictive because those are the cells lining these small areas, the ventricles and the canal. The central canal, that is. An important note on microglia. So all right, so here's the little chart I made that has pretty much everything you need to know about the cells of the CNS. I pretty much just took that diagram that's on page 11 and expanded it and added all of the words you need to know. So a lot of this is if you know the keywords that are listed and you can word associate that, you should be good. So one thing I do want to mention as well, while you're studying, and like I said before, a good fill in the blank question would be, what are the derivatives of the glioblasts? And then you would write in astrocytes and oligodendrocytes. So while you're studying, you should be thinking of ways that these questions can be asked in the exam. That's like my best advice for studying for the fill in the blank portion. Everything you look at, you should be thinking, okay, how can he ask this as a multiple choice question? How can he ask this as a fill in the blank question? So just looking at the diagram, you can also say something like, how can he ask something about the neuroepithelial cell derivatives? He could say, which of the following is not a neuroepithelial cell derivative? Or he can ask, write out the cells that are neuroepithelial derivatives. So looking at the diagram, it would be, the answers are neuroblasts, ependymal cells, and glioblasts, because those are the 3 cells that neuroepithelial cells differentiate into. So he could ask that as a multiple choice by saying, which of the following is not a neuroepithelial cell derivative? Your answer, it could be A, neuroblast, B, ependymal cell, C, glioblast, D, microglia. Microglia would be your answer, right? Because those are down on the left. Those are not, neuroepithelial cells are not on the diagram. They are actually mesodermally derived. I'll type out that question I just said so that you guys can see it all in writing too. So I'll do that on the next slide. Again, let me know if you have any questions. I'm not covering everything with the histology stuff. Here's a question I kind of talked through on the last slide, so you guys can actually see the words there. Take a screenshot of that diagram I posted that I made. It's in my handwriting. If you want to write it out, do that, but that's a good resource for studying the histology stuff. All right, so moving on to a new topic. I'm on page 14, number 8. Neuron classification. So there's two ways we're going to talk. There's two different ways you can classify neurons, anatomically versus physiologically. What you need to know is anatomic refers to the appearance, physiologic refers to the function. So we'll talk about those in the next page. So page 15, we're starting off, we've got unipolar, bipolar, and multipolar. So if you're taking the prefixes there, we've got 1, 2, and multiple, right? These are the anatomical classifications because we're talking about how many processes there are, what it looks like. So first one is unipolar. It's called unipolar because it looks like there's only one process, but there's actually two. So that's why if you look on the right here, I kind of tried to draw them out. It looks like it's just one coming out, but there's actually an efferent and an afferent. So that's why it's also called pseudo-unipolar. So that's what it looks like. And you have to know where you can find these types of neurons. So these are going to be your general sensory neurons. When you think general sense, think touch. Think ganglia, think DRG. And the reason for this, and I mentioned it earlier in Unit 1, dorsal root ganglion, it's coming back again, that's where I have DRG. Those are the groups of cells that carry sensory information to the CNS. It's nice too, I'm all about when letters line up nicely for us. We've got general starts with a G, we've got ganglia starts with a G, and we've got DRG. So whenever I read unipolar, I would think general sensory, ganglia, dorsal root ganglion, DRG. Next we have bipolar. So two processes, and if you look to the right, it looks like you can tell there's one going towards and one leaving. There's one afferent and one efferent, which is actually the same as unipolar, just the unipolar looks like one. So where you can find bipolar neurons is going to be ones that deal with the 5 senses. So your special senses, smell, vision, hearing, taste, equilibrium. Next, we've got multipolar. So we've got multiple afferents going towards, as you can see on the little drawing on the right. Multipolar are going to be found in our motor neurons. So I've got in red there, multipolar, motor, most common, they all start with M. So every time I was studying and I got to this multipolar part, I would just say it out loud, multipolar, motor, most common. They all start with M. That's a quick way to remember that, because you do have to know that they are most common and that they're found in motor neurons. You've also got to know the difference between Golgi type 1 and Golgi type 2. These are just differentiating whether there's a long or short axon. So it's kind of, I feel like, tedious memorization, but Golgi type 1 are going to be our long. How an easy way, quick way for me to remember that is the 1 looks like an L. So 1 is long, we've got 2 is short. So make sure you're aware of that. That was how I remembered it for the exam. And then I've got this red box down here. So I just want to point out every neuron only has one axon. We're only going to have one efferent process or one leaving. So this anatomical classification, we've got multipolar, meaning multiple processes, but that's multiple because we've got multiple afferents coming in. So we don't have multiple efferents. Every single neuron only has one axon. So I wanted to point that out. Just as a reminder down here, I've got remember same, so sensory is afferent, motor is efferent. And if you forget that an axon is an efferent, just remember the axon is going away from the cell body. Axon moves away. Moving on to physiological classification. So this is based on their function. On page 15 at the bottom, we're starting off with sensory. So that's sensory information is what's carried towards the CNS. So we've kind of been talking about that since the beginning. That's our afferents. On the next page, 16, moving on to motor. So that's information going away from the CNS. Again, talked about this already. So that's our same. Sensory afferent, motor efferent. Then we have internuncial neurons. So internuncial neurons are neurons that are entirely within the CNS. So what is our CNS? The brain and the spinal cord. So these neurons never leave the brain or the spinal cord. There's a few different subdivisions of internuncial neurons. That's the rest of the slide, the rest of this page 16. So we'll start with commissural. Commissural neurons are what connect equivalent structures. So the example Dr. Torgerud puts in the book is left precentral gyrus to right precentral gyrus. So if he gives you an example on the exam and it's going from one side to the other to the same exact structure, the same word, then that's going to be your commissural neuron. Next, we've got projection neurons. So these are ones that begin in one structure and end in a different structure. So how I think of it is what does project mean? It means to extend out, right? So I think extending out to another structure. Now we have two different ways we can classify projection neurons. It can begin in one structure and terminate in a different structure on the same side of midline, or it can cross midline. So the words we use to determine that are ipsilateral versus contralateral. Ipsilateral means it's staying on the same side. Contralateral means we're crossing to the opposite side. So he'll probably ask this on the exam rather than asking for a definition. He'll give you an example and say, what kind of neuron is this? So if we have an ipsilateral projection neuron, the example we're using here is right side of the cerebellum to the right side of the pons. So looking at that example, I know it's not a commissural neuron because it's not going from the right side of the cerebellum to the cerebellum again, because commissural connects equivalent structures. So I know it's projection because it's going from one structure to another. And then looking at the side of midline, we have right side to the right side. So that's same side, making it a projection ipsilateral neuron. The next example under contralateral, we're looking at right side of the cerebrum to the left side of the midbrain. So if we're going from the cerebrum to the midbrain, those are two different structures, two different words. So we know it's a projection neuron. And then we look, it's going from the right to the left. So it's crossing midline. That makes it a contralateral projection neuron. All right, and the last part of this section is going over the intra and intersegmental neurons. These are specific to the spinal cord. Internuncial ones that don't leave the cord. So if you think about, I compare this to highways. If you have an interstate highway, that is a highway that goes to different states, right? If you have an intrastate, it's a highway within the same state. So applying that to these terms here, if you have an intrasegmental internuncial neuron, it's staying at the same cord level. Whereas if you have an intersegmental internuncial neuron, it's traveling to a different cord level. So I have on the right here this little image with T6 and T7. We can apply ipsilateral and contralateral to these as well. So starting from the top, I've got that it says T6 and there's this tiny little neuron going from part A to part B. It's on the right side of midline, so it doesn't cross midline, making it an ipsilateral, and it's staying at the T6 cord level, so it's intrasegmental. So it's an ipsilateral intrasegmental. Jumping down to the next one, So we've got one that's traveling from T6 across to the other side of T6. So that's contralateral because it's crossing sides, but it's not leaving T6. So it would be contralateral intrasegmental. Then we've got the dotted line one that's going from T6 down to T7, but it's crossing over to the other side. So that's a contralateral intersegmental. We have and the last one that goes straight down from T6 to T7. It's staying on the right side, so it's ipsilateral because it's not crossing midline. And it's going from T6 to C7, so it's ending at a different chord level, making it contralateral intersegmental. So again, you'll probably see these as examples on the exam. He'll give you an example of where the neuron is going from point A to point B, describing it, and then you have to label it what type of internuncial neuron is. And lastly, this is my dog Charlie. He's been staring at me talk to the computer the whole time, so just wanted to share that with you. So this video is going to be part 1 of Unit 2. I want to get this up as quickly as possible, so I'm going to post this now, and then I'll work on the last end of Unit 2 and get that to you guys hopefully by the end of today, Friday. So as always, let me know if you have any questions. Hope you guys found this helpful. Thanks.